Happy New Year and welcome to the Diabetes Bio Podcast, a podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. Each month, we will interview one or more authors from the feature articles, the papers of the month, or other articles published in that month's issue of the Diabetes Journal. We also have a Sweet Talk segment of the podcast, which is our open format portion of the podcast that can include topical interviews or discussions. My name is Darlene Sandoval, and I am joined by my podcast co-hosts, Dr. Kirk Habiger and Dr. Kevin Williams. How are you guys doing today? Happy New Year's.
Yeah, Happy New Year, Dar. Happy New Year, Kevin.
Yeah, Happy New Year to you too.
So I have one question for you. Who stayed up till midnight on New Year's Eve?
Yeah, I unfortunately was awake.
Yeah.
Both at the East Coast and the Central Time.
Nice. Kevin?
I was not awake, and maybe I'm a little ashamed to admit this because I haven't admitted it yet, that I sent out Happy New Year's text to several people, but I scheduled it. So I scheduled it for midnight, and I could be in bed asleep.
That is great. That is amazing. I am really proud of myself. We celebrated at the Eastern time zone. I'm in the Mountain time zone. So we celebrated at 10 o'clock Mountain time. But then we actually ended up staying up till midnight. I was really surprised.
Oh, wow.
I expected I was going to go right to bed, but.
So, I mean, I guess as a follow-up question, Dar, how many of the artists did you recognize on the New Year's Eve celebration telecast?
Yeah.
Right? I mean, every year it's less for me.
Yeah, exactly. Exactly. All right. Well, so this month we have a very special episode. It's basically an extended version of our Sweet Talk segment. So Kevin, why don't you tell us about it?
Yeah, we broke this into a two-part Sweet Talk segment. In part one, we're speaking with two pioneers in diabetes and obesity research, Dr. Philipp Scherer and Dr. Joel Elmquist. They're both from UT Southwestern in Dallas, Texas. Dr. Scherer and Dr. Elmquist are previous recipients of the ADA's Outstanding Scientific Achievement Award, and more recently, Dr. Scherer received the ADA's Banting Medal. Both have contributed significantly to the society, including serving or currently serving on the ADA Pathways Award Committee, which has been so critical for the funding mechanism for so many early career scientists. So they're here to share their perspectives on this award, the evolution of diabetes as well as obesity research, the interplay between the brain and adipose tissue, and what the future holds for understanding and treating these complex diseases. Kirk, tell us more about part two.
In part two, we're going to have an interesting discussion to continue the conversation with the recipient from an ADA Pathways Award winner, Dr. Jonathan Flak. He'll be discussing his funded work on the brain's role in glucose regulation, energy balance, and body weight control. It's a great segue from part one's discussion with Dr. Elmquist and Scherer.
All right, well, let's jump in.
Today, we're thrilled to have with us two prominent researchers in diabetes and obesity research, Dr. Philipp Scherer and Dr. Joel Elmquist. Dr. Elmquist is the Carl H. Westcott Distinguished Chair in Medical Research and holds the Macklin Family Distinguished Professorship in Medical Science at the University of Texas Southwestern Medical Center in Dallas, Texas. He was the recipient of the ADA's Outstanding Scientific Achievement Award in 2014 and has made groundbreaking contributions to understanding how the brain regulates both energy balance and glucose metabolism. Dr. Scherer is the Gifford O. Touchstone Jr. and Randolph G. Touchstone Distinguished Chair in Diabetes Research. He also holds the Touchstone West Distinguished Chair in Diabetes Research at the University of Texas Southwestern Medical Center in Dallas, Texas. He was the recipient of the ADA's Outstanding Scientific Achievement Award in 2005, and more recently, the recipient of ADA's Banting Medal in 2015. He is renowned for discovering adiponectin and revolutionizing our understanding of adipose tissue biology. Thank you both for joining us today.
Glad to be here.
Yeah, happy to be here.
So before we get into the larger discussion, we always like to start off with a little bit of an icebreaker question. And the question we have is, if you could solve one unanswered question in diabetes or obesity research today, what do you think that would be? Dr. Scherer, we can start with you, and then we'll move on to Dr. Elmquist.
I think one of the biggest unresolved issues that I would love to have more insight in is this famous concept of the lipostat, or whatever it is, whether you believe in the lipostat or not. There is some mechanism in place that really freezes our weight basically at our maximum weight that we've ever achieved throughout life. And we still lack pretty much any deep insights into what controls this lipostat. Is it centrally regulated? There's certainly a possibility. Or is it perhaps, in a more heretic hypothesis, peripherally regulated by tissues such as adipose tissues? So I think that's still one of the biggest questions, and it's particularly relevant now in the context of weight loss drugs, where we know you get off the weight loss drugs, you bounce back to your original weight. So
Yeah, it's a fascinating question there. Dr. Elmquist?
Of course, there's many things I don't understand, but one that I still am puzzled by, and we continue to try to work on this, is it's quite clear that several hypothalamic nuclei circuits are really good at regulating metabolism independent of body weight, and the most obvious being a hypothalamic control of liver metabolism. And to this day, I really don't understand how manipulating, let's say POMC neurons in a number of ways acutely affects liver insulin sensitivity, glucose production, lipid production, etc. And we really don't know and have a good understanding why or how that is regulated. And so I think, to me, that's still a very important question to address, because, I think, as Phil mentioned, in a heretical way, that I think one of the first lesions in type 2 diabetes is something that gone awry in the hypothalamus, which leads to insulin resistance in the liver. And so I really would like to understand that better.
It leaves a lot of work for us younger scientists to go after.
That's it's also, you've set the stage very well for my next thought, which was assembling this podcast is really exciting. Here we have two experts from the field of neuronal control of metabolism, and one very much central and the other more peripheral. And so we're going to be able to pick your brains here on the relative importance of those to diabetes and obesity research. But before we get too far down that road, maybe we can start with a bit of reflection. You both contributed to the field during a period of tremendous scientific progress. So taking a step back in time, can you maybe provide some insight on when you both entered the field? What was the landscape like, and how you've seen it evolve over the years? And we'll start with Dr. Elmquist.
Yeah, thanks. It was exciting times, and there were at least three major discoveries that were almost simultaneous that I often think about, and I talk about when I'm teaching graduate students and medical students. One, of course, was the discovery of leptin by Jeff Friedman's group in December of 1994. Then there was the cloning of the melanocortin receptors and understanding the role of the melanocortin system by Roger Cohn, Kathy Mountjoy, who's a colleague of mine and was a postdoc with him at the time. Very fundamental. And then there was work Phil was doing across the river from us when I was at the Beth Israel, when he discovered another critical adipokine, adiponectin. And Phil would have to remind me of the exact timing for all three of those, but they were almost simultaneous. And it really changed the field. And it was literally that they were like thunderbolts that drew a lot of people into this area of research from very disparate backgrounds. So it was really, really exciting in that regard.
Yeah, that's great. And Phil?
So obviously, Joel and I come from the same generation. So we're going back now 30 years, basically, early mid 1990s. And Joel summarized it very well. It was an exciting time. For me, as a postdoc, I entered the field at the time, and this was really when metabolism research was mostly focused on 3T3L1 adipocytes and the translocation of GLUT4 to the plasma membrane in response to insulin. And you start your postdoc time and you think, what's my niche going to be? Because there were so many groups intensely focusing on that aspect of sort of the cell biology of the fat cell, that it seemed to me that this is probably not the way to go because the competition seemed tremendous. And that's when I sort of made the decision, maybe we should be focusing on secretory proteins from the adipocyte, the secretory apparatus, other aspects of the adipocyte. And this goes way, way back. And this was indeed the time when that shift occurred. And then, of course, the cloning of leptin. We were kind of going hand in hand. Our publication on adiponectin was about a couple months later, but this all basically happened at the same time. And we were concerned, in fact, for a time that we might be chasing after the one and the same thing between the Friedman lab and the Lodish lab at the time with adiponectin. But I think the fat cell has come a long way since then, and certainly its interaction with all these other tissues, including the brain. But the landscape back then was much more cell biological, less whole body physiological. And it seemed very, very biased towards just one process. So there was plenty of room at the time, and clearly it's given us 30 years plus of research and still expanding in terms of the possibilities as to what the fat cell can do for you.
All right. So my question is for Dr. Elmquist, and we're going to fast forward a little bit. I think many of our listeners might still think of brain control of glucose now. So we're switching from energy metabolism to glucose as more of a response to stress, right? Autonomic responses to stress, regulating hepatic glucose production, et cetera. But I think your work has contributed to thinking of the brain as sort of a glucostat that's looking at regulating glucose under a variety of conditions, including postprandial conditions. So can you talk a little bit about these processes and maybe why you ventured into this area of research?
Yeah, thank you. To answer your second question, why I kind of went into that area, one, it fascinated me that you could really, at least in mice and genetic models, dissociate changes in body weight from regulation of glucose homeostasis. But the main reason was that the whole field was going towards a certain area investigating feeding and body weight. So I've always tried in my career to kind of go away from the herd, if you will. And so, for example, one of our earlier papers was led by a postdoc at the time, Roberto Capari, working with Brad Lowell and I. And what we found was that replacing leptin receptors in the arcuate nucleus using a viral approach had very modest effects to rescue the obesity in that model. It was a leptin receptor null model that could be reactivated. This is a mouse made by our colleague Streamson Chua. Just to cut to the chase, what we found was putting back leptin receptors in the arcuate nucleus, the famous nucleus where the POMC and AGRP neurons live, had very modest effects on body weight, but normalized the dysregulation of glucose. So essentially cured their diabetes. And so it to me was just like a eureka moment that you could really find a hotspot in the hypothalamus where leptin could regulate glucose homeostasis in an otherwise obese animal. So that's why I started down that road, is because of those original findings. And that was long enough ago. That was the very first issue of Cell Metabolism.
Cool. And so do you think the brain is a target for diabetes treatment?
Oh, for sure. And I think we're living in that area right now, in that, our colleagues in GLP-1 space, including you guys, all of you on the call, it's clear that many of the actions of GLP-1 drugs, and of course on obesity, but I think the anti-diabetic actions as well were central. We had a paper back in the old days with Drucker hypothesizing that.
You both have served on the advisory committee for the ADA Pathways Award, which is a program that supports innovative research from early career scientists. And we've had several of these amazing recipients as guests on the program over the last year. We've also had Dr. Christopher Newgard and Dr. Lewis Phillipson, who both chair this committee on our program. I was wondering, for you who have been a part of this program, what has it meant to be a part of this program? And how have you seen it evolve over the years? Let's start with Dr. Scherer and then move on to Dr. Elmquist.
Well, I was fortunate to be part of the very first incarnation of that program for the first couple of review sessions. And that was clearly a very great privilege to be part of it in terms of really the ability to review grants from the top researchers at every universities in the area of diabetes and obesity research. So it's been quite amazing at the time, and it's perhaps also programmatically, it's been extremely rewarding to actually watch the success of the recipients of these awards as it has advanced their careers. And many well-known names now that we read papers from are going back to being recipients of this particular award. So I congratulate the ADA for having the idea of putting a lot of eggs in few baskets, which it was controversial at the time, basically, because of course it comes at the expense of giving a broader range of grants to more people. But I think it's really paid off, and it's been a really good investment.
Dr. Elmquist?
Yeah, I completely agree with what Phil says. I joined the panel probably because Phil was rotating off, but I've been involved in the past several years, and it's been great. And the thing that really strikes me, and Chris and Lou and others on the panel and Mike Schwartz, people like that are great examples of this, is it's showing that leaders in the field, senior folks, are very interested in the next generation of diabetes research. And, I think this program exemplifies that. And the reviewers across the board are very supportive of these younger investigators. And it's really fun to watch the panel work, but also to see the fruits of the funding for our younger investigators.
I know I personally agree totally with both of what y'all have said. And the journal Diabetes has made an effort to highlight the award winners so that they come back and write articles for the journal to discuss what their research was funded on and the progress that's been made on that. In our Diabetes Bio podcast, we've had, as I mentioned, several of these recipients on, and it's really been neat to see how the work was thought of, created, developed, the progress of that work and where it's gone even after the ADA Pathway Award had ended, and it's moved on to other forms of funding, NIH funding.
Yep.
And it's been a really successful program. So it's really credit to you guys being on that committee and the chairs of those committees for Dr. Newgard and Dr. Phillipson to really drive that program. So it's really nice to see.
Yeah, that's great, Kevin. And I would like to add that Dr. D'Alessio is actively involved with the review as well. And now with his editorial role, I think that's a great idea. So kudos for that.
It's a really neat award because it feels like it supports a little bit more on the innovation side, or a little bit more
Yep.
of the riskier science, right? And so it's cool to interview some of these folks that won some of the earlier awards and see what kind of progress they've made and what kind of impact they've had, actually.
Absolutely. So I want to take a little bit of a left turn here. And Phil, was hoping you would maybe give us some thoughts on adipose tissue, right? So for many years, fat is primarily viewed as a passive energy reservoir and often thought of as bad, right? Muscle good, fat bad. And we know, based on your work and others, that it's a lot more nuanced than that. So what can you tell us about this evolution in thought, right? From passive reservoir that may be harmful to possibly healthy and a very large contributor to our overall metabolic health?
Obviously, work from many different labs have contributed to that. And I think there is now overwhelming evidence that adipose tissue has exceedingly beneficial roles for the overall metabolic well-being of the system. And that really relies on every level of regulation, whether that's at the proteomics level, at the lipidomics level, at the metabolomics level. So adipose tissue serves as a great source for many of these beneficial factors. And it does, of course, get stressed when we put too much pressure on it in the form of excess calories coming in. And all of these things change in response to that. So it's been incredibly insightful to see this over the years, how that changes. But perhaps there are a few things that illustrate better the power of adipose tissue, which is something that people know about but pay very little attention to. Is we noticed over the last couple of years in a number of different models that we only have to transplant a small amount of adipose tissue in the form of 100, 200 milligrams that we have manipulated in one way or another, in a positive or in a negative way. And we can convey a phenotype upon the recipient mouse by giving that mouse a little bit of adipose tissue. And that's just remarkable, because it shows you that there is tremendous potential at that level. And we don't have to manipulate fat tissues across the board. It takes relatively little to convey these positive effects on the recipient. So that's something which keeps fascinating me in terms of the magnitude of the effects that you can achieve. And it does hint at many more things to come from adipose tissue physiology in the future.
Yeah. And I guess as we talk about, what does it hint for in the future? Where do you think maybe future therapies will act, especially within the adipose tissue? Is there a role there? We spoke a little bit about, the brain and GLP-1s, but are we going to have specific therapies targeting adipose tissue or adipocytes?
Increasingly so. So this is not restricted to just secretory proteins that you can either have in a recombinant form and inject, or that you can counteract with neutralizing antibodies. That's equally important in terms of the negative players. But it's also apparent to me that, now we are sort of entering also a stage of, I guess, gene therapy in the widest sense. AAVs, adeno-associated viruses, become increasingly popular as clinically relevant interventions, not just for gene repair. And I like the idea that we have a lot of, superficial subcutaneous fat easily accessible from the outside to these AAVs. So I think there's a lot of potential there in terms of adipose tissue manipulation, adipocyte manipulations, not just restricted to AAVs, but also increasingly we see strong evidence for interventions at the level of antisense RNAs and microRNAs that can be targeted selectively to adipose tissue. So huge therapeutic potential there.
So back to the, I would say, broader impact of both of your work, right? So you have this impact certainly on advancing our scientific knowledge, but also you have trained many scientists behind you. And Joel, we can, whether you want to take credit for my podcast co-host or not, that's up to you. But if you had to think about what was one of the key skill sets that is required to be a good mentor, what do you think that would be? We'll start with Dr. Elmquist.
Yeah, I've been really fortunate through my career to have a lot of really smart, brilliant trainees, including our co-host, Dr. Williams. And this goes
Say that
back
again?
to
It is recording, right? Quiet. We got it recording.
It goes back to my own postdoctoral mentor, Cliff Saper. And Cliff, to this day, is a very busy guy. But when I was a postdoc with him in the Department of Neurology at Beth Israel and Harvard, he was chair of a clinical department, chair of the neurology department. I think the first year I was there, he was the chair of the entire Society for Neuroscience meeting, which is a giant job. And he ran his lab in the way that he gave us a lot of freedom, but was always there if we needed him. So I would email him and he would make time, and we would sit down often at the microscope, and which was an amazing thing to do. So I've tried to model my own career and mentoring style in that regard. And yet the basic thing is you recruit people that are smarter than you, have the resources for them to do the work, and then get out of the way. Give them enough freedom that they can use their own creativity and intellectual curiosity to come up with things that you never would have thought of. And it's worked well for me, at least. I've been fortunate to work with a lot of really talented people through the years.
Awesome. Dr. Scherer?
Well, I think Joel summed it up very well. I think we have a very similar philosophy in the sense that we like to be part of projects. We like to give advice, but maybe try to avoid micromanaging people. I think things always work best when you let them loose, give them a very long leash and let them go where they want to go, as long as you can responsibly sort of do that under the big umbrella of whatever research that we are covering. Good news is when you work on adipose tissue, everything is game, because everything at the end of the day boils down to adipose tissue again, whether you go to the liver, to the brain, or to the immune system, the kidney, it's always the adipose tissue at the end. So basically giving enough freedom to really be creative, get out of the way, and to surround yourself with people that are basically smarter than us is definitely the key. The other aspect, I believe, which is very important, I think, is just also to some extent lead by example, to really never ask anybody in the lab to do something that I wouldn't do myself. And I think it takes a lot of hard work, and you have to be engaged and you have to be excited no matter how many years you've been doing it. You have to stay really excited about it. And that excitement that you feel, I think, transpires very well down to the people that work, the colleagues that work with us, around us. And I think that's part of the secret.
Darlene, if I could, I just add one thing
Absolutely.
that I forgot. The thing that Cliff Saper instilled in me was how important it was to write and be involved in the writing process. That includes fellowship applications, but also the papers. And so I really tried to instill that through my career to all my trainees. It's financially, it's important that they get the fellowship, of course, but I think it's more importantly required training to survive in
Yeah.
our business. And so that's one thing that I've always tried to incorporate as well.
So on the flip side, to follow up on that, what key piece of advice would you give to young scientists that want to make an impact on the field?
Don't overlook how important communication skills are, whether it's giving a good talk, your slides aren't a mess, to writing. And if you cannot do those two things in biomedical research or academic medicine, if you will, then you're not going to be as successful as you can be.
Bill?
Yeah, I think that's definitely important. I think it does take hard work. That has not changed, whether that's our generation or the future generation. You have to really commit yourself to it, but at the same time, you have to find that delicate balance of being on it all the time while not burning out. And there is increasingly, of course, also this emphasis on kind of life-work balance. And it is a matter of juggling a lot of different things. But at the end of the day, I think the people that are really vested emotionally in the science are really the ones that succeed at the end of the day. And everything else comes more or less. If you're excited about what you're doing, everything else comes automatically. So best thing we can do here is to provide an environment where people are able to do what they want to do. So we basically try to provide a sandbox, which has a lot of toys in it, and then just let people play in there. So I think that's the key.
Thank you for that. That's great.
Yeah, great analogy. So as we wrap up, the field of diabetes and obesity research is constantly evolving. We started off the podcast today with a retrospective look at the landscape when you started your careers and what it was like and how it's evolved over the years. Looking forward, where do you see the field heading in the next 10 years? Do you see any emerging technologies, therapeutic strategies, or areas of research that you're particularly excited about in moving the field forward? Let's start with Dr. Scherer and then move on to Dr. Elmquist.
Well, I think we live in the best of all times right now, because it's clear with the weight loss interventions, to me, where we now have such a profound impact on the liver, on the kidney, on the cardiovascular system, heart failure. And the vast majority of these effects rely critically on weight loss. There's some wrinkles in that, of course, and not everything relies on weight loss, but a lot of it relies on weight loss. So that really opens the door again to more efforts in the area of adipose tissue physiology, a better understanding of what the adipocyte has in it, what can it do for us, finding ways by tickling it the right way, such that it is actually, rather than that analogy I always use, the angry adipocyte that is inflamed and fibrotic, we want to make it into a player to the benefit of it all. And I think given the technologies that now are at our fingertips in terms of manipulating things in a relatively cell type specific manner, including in the adipocyte, I think the next 10 years will give us a lot of opportunity to be a lot more creative than we used to be in terms of thinking about clinically relevant manipulations that we perform in the preclinical models.
That's great. Dr. Elmquist?
I think Phil highlighted an important point about the obesity drugs and what they are doing and potentially give us a chance to do going forward. And I return to this idea of potentially segregating the effects of body weight and weight loss per se from other things, including the metabolism. But, the hypothalamus controls complex behaviors as well. And there are these really interesting anecdotal reports of intersection of circuits that control body weight, but also that control things like mood, rewarding behavior, even addiction. So I think given the revolution of the omics as well as, optogenetics and chemogenetics, I think there will be in the next 10 years the ability to start to tease apart how the hypothalamus regulates all these endocrine behavioral and autonomic effects in a manner that's independent of weight loss per se.
Yeah, it's amazing to think back years ago being able to do some strategy like that, and now today how it
Yeah,
could become
no
so
doubt.
much more straightforward. Thank you both, Dr. Scherer and Dr. Elmquist, for joining us today, and thanks to our listeners for tuning in to this section of the Sweet Talk. Dr. Scherer and Dr. Elmquist, please tell the listeners what you have coming up, or maybe where they can find you and learn more about your work.
Well, I guess we both have a website that can be tapped into with and Twitter X sites that can be easily found out there to get a sense of what's going on locally here within the Touchstone Diabetes Center and the metabolism group at large here, including our NORC that glues us all together, the NIDDK-sponsored obesity center. So we are a tightly integrated group here. It's been a privilege to be here with Joel for the past, 18 years at UT Southwestern. And I think it's a classical example of how we both have our favorite tissues and cells, but it's really a teamwork that allows us to integrate a lot of these metabolic issues into an interplay between different organs, including fat tissue at the brain.
Yep. And I would just add, yes, look at our center website that Dr. Williams does such a great job of keeping relatively up to date. I think it's hypothalamus.edu, center. And then, yes, we should mention the NORC again, that, you can check out the website there, and it's to highlight what Phil said or amplify, it's really an amazing group of people here. And, the NORC led by Dr. Jay Horton, that Phil and I are co-directors, I think is kind of a way to tie everything together. So yes, I encourage you to check out those websites.
That's great. We highly recommend checking out more of what Dr. Scherer and Dr. Elmquist are doing. And as always, please like, follow, and subscribe for more diabetes bio content. Welcome to part 2 of Sweet Talk. I'm Kevin Williams.
I'm Darlene Sandoval.
And I'm Kirk Habeger.
Today, we're joined by Dr. Jonathan Flak from the Indiana University School of Medicine, a 2016 recipient of the American Diabetes Association Pathway to Stop Diabetes Award. Dr. Flak's work focuses on the ventromedial nucleus of the hypothalamus, or the VMN, and its role in glucose regulation, energy balance, and body weight control. His research has provided insights into how specific neuronal populations contribute to metabolic health. He has also contributed a perspective on his Pathway Award in this issue of Diabetes, which we're excited to discuss today. Welcome, Dr. Flak, and thank you for joining us.
Thank you all for the invitation to join you today.
John, we'd like to start these things off with a little bit of an icebreaker question. If you were not focused on neuroscience and metabolic disease research, what other field or career could you see yourself pursuing?
Yeah, so that's an easy one for me to answer, because actually I went to school for something completely different than research. When I entered undergraduate studies, I had planned to be a clinical psychologist. I was very interested in the brain, and that was the natural place in which it was logical to continue my studies, and that was a profession that you could do and continue through psychology studies and investigate. on that topic. However, eventually I realized that that wasn't the right path for me. And at the same time, I had been very involved in research and had gotten some encouragement from the PI that I was working with to pursue graduate work, which led me on the career path that I'm on.
Nice.
John, first of all, congratulations on the ADA Pathway Award. And I was hoping you could start by giving us an overview of the ventral medial hypothalamic nucleus, why it's so important for energy balance and glucose homeostasis, and then maybe touch on some challenges that we have in studying the neurons of this structure.
Yes, that's a really good, great place to start. So this area of the brain, I think overall, it's been tied. I think the main through point to the function overall of the nucleus is really in terms of driving the sympathetic nervous system. So there's functions within the nucleus that can promote glucose clearance, as well as those that can promote mobilization of glucose, and those are really intermingled throughout the nucleus. That has been a challenge to study the nucleus as a whole, because you have mechanisms that can increase blood glucose and decrease blood glucose essentially intermingled throughout the whole area. So it's kind of the idea in terms of you're studying the pancreas as one individual thing rather than studying alpha cells, beta cells, delta cells, and all these. Unfortunately, in the brain, we're really behind a lot of these other tissues and organs in order to understand what are the different subpopulations and different markers of different cell types. That has really been, I think, an obstacle in order to investigate a lot of these structures in terms of the individual functions in different physiologies, behaviors, because I just mentioned, for example, just the aspects in glucoregulation. But in addition, that makes it even more complicated is that there are important functions in the ventral medial hypothalamic nucleus in terms of energy expenditure control and behavior responses and reproduction that really make it challenging in order to really find ways to target the individual functions without just affecting the nucleus as a whole. Hopefully within recent years, especially through some of the work with single cell sequencing and some of the work coming from different groups in studying individual subsets of cells using Cree mouse lines, hopefully we can get better to disentangling some of the specific functions by different portions of this nucleus. But as I state that's not only a problem with an area such as the ventral medial hypothalamic nucleus. There are places all through the brain. The brain, unfortunately, the beauty of it is that it's so complex, and there are all these unexplored avenues that we can hopefully eventually specifically target someday. But the difficult part is that there's so much left to understand in the specific functions.
Yeah, that was great. And I love the analogy back to the pancreas, right? It has multiple functions if you just consider it a single structure, but that's not what it is.
Absolutely.
Let's layer some more complication on this. You talk a little bit in your article about the sexual dimorphism in this nucleus as well. Can you talk a little bit about that?
Yes. So this is definitely a structure that has a lot of sexual dimorphic descriptions in particularly in terms of the responsiveness to estrogens. That has been heavily studied in recent years from groups like Holly Ingram. Yang Shu have really been able to disentangle some of the specific estrogen mediated functions within the nucleus. And there are certainly specific reproductive functions that come from different subpopulations within the nucleus. That is certainly another level of complexity that in order to understand how different sex hormones regulate the function of these individual aspects of VMN related physiology and behavior.
So, when we talk about the complexity here, how optimistic are you that there could be strategies to target the brain in therapy for obesity or diabetes?
I mean, I'm hopeful that there's some way in order to target these things, but as of now, we don't have specific mechanisms that we can target. That if we're allowed to utilize the same kind of techniques, I would imagine in the rodents that we can turn on different pathways and turn off different pathways. But realistically, we would need some kind of small molecule or peptide agonist to target those pathways. That being said, in terms of particularly, I can think of especially on the energy expenditure front, there are avenues in which to target potentially those pathways in combination with those that suppress food intake that could be in the future useful. However, these specific pathways, we don't have those mechanisms yet. However, given some of the strategies of combining incretin agonists with things like melanocortin agonists, that could be on the horizon eventually once we get to identify these specific markers that can mimic some of the strategies that we employ in terms of turning on or turning off these pathways. I think it's possible, but we're unfortunately just not there yet.
I think for our listeners that are studying diabetes, they're very interested in understanding the issue of patients that undergo hypoglycemia unawareness. And in your article, you do touch on the VMN's role in counter-regulatory responses to hypoglycemia. I was wondering if you could explain to the audience how the VMN neurons do this. How do they sense glucose? How are they responding to low glucose levels? And why the system is critical for preventing hypoglycemia?
Yes, that's a really important fact, and the focus of the Pathway Project was really in trying to understand the pathways downstream from the ventromedial hypothalamic nucleus that can mobilize blood glucose in and protect us from life-threatening hypoglycemia. And certainly, this is a problem that's been known for a long time. That following recurrent episodes of hypoglycemia, your endogenous responses that normally protect you from hypoglycemia dampen over time following these recurrent episodes. And so it's known that there are these pathways that just for some reason get turned off over time, and we're able to measure that in the lab through these CCK-BR cells within the brain. So they receive these signals that there's low blood glucose, and what they do is they exert these responses that can potently increase blood glucose. And I was definitely quite surprised in lab the sheer magnitude of these effects, and that I had animals that had readings of blood glucose that were above what was even able to be registered on the glucometer. And just by activating these cells and likely this pathway throughout the brain. Now, what is the exact signal that they receive that response? I would imagine that they're glucose inhibited cells. There's known to be glucose inhibited neurons within the ventromedial hypothalamic nucleus that then, when glucose levels are too low, then they become activated and they drive this neurohormonal response that then mobilizes blood glucose from your energy stores and then increases blood glucose in the context of that hypoglycemia to again protect you from life-threatening levels of hypoglycemia.
John, we focused a little bit here on glucose, but I wanted to give you an opportunity to touch on the role of the VMH on adipose and thermogenesis. So maybe you could expand on that a little.
Yes, so that was something that was really a surprise. Was not a primary project that I had from the Pathway funded project, and it came out of studying a neuropeptide that was expressed within a subset of cells within the ventromedial hypothalamic nucleus, and actually at the time thought that they were driving this counter-regulatory response, again, this protective response from life-threatening hypoglycemia. However, when I knocked out this neuropeptide, there was actually no impact, at least in my hands, on counter-regulatory responses or blood glucose really in general, other than the deposition of fat, increase in body weight. And then that led to a project that shot off from this main hypoglycemic project, in which we're starting to understand what are the neural pathways that can drive energy expenditure. And so what we're able to do is to see that these projection goes downstream into this area called the preoptic area. The preoptic area has classically been known to be very important in thermal regulation. So it makes sense if there are cells that would be engaged, maybe not in necessarily directly for thermal regulation, but maybe pathways that would utilize similar mechanisms to increase energy expenditure, catabolism of adipose tissue that would happen when you activate them. So that was a separate project, and we're very interested in understanding the underlying mechanisms of that. We've been able to tie it to generating a thermogenic response in the both the brown as well as the white adipose tissue. But exactly the underlying mechanisms in different organ tissues, we don't know yet if it's something that maybe more integrates multiple tissues together to then target the overall catabolism of excess tissue, or this is something specific to adipose tissue control. That's something we're continuing to measure.
And John, looking ahead, what do you think is the most exciting unanswered question about the VMN, or maybe in more general terms, in the study of obesity and diabetes that maybe your lab is focusing on?
Yeah. So one of the things I think is just the specificity of how all of these different singles are intertwined. So there's some evidence that's been coming out from other groups that it's not necessarily just one set of cells that controls responses to hypoglycemia versus those that drive hyperglycemic responses, but there's very targeted pathways for individual components of that. And what I mean by that is that there are some pathways that inform the liver versus others that regulate pancreatic function versus those that impact things like adipose tissue. Now, we'll see in future years whether that's the case. There are these individual circuits and individual downstream components, or is it a merely one set that has been the previous model that integrates that information across multiple tissues? So, for example, in terms of glucose clearance, perhaps there are some that suppress impacts at the level of the pancreas and the liver, as well as those that drive clearance at the level of the muscle, or are those separate functions, separate sets of cells that govern each one of those responses? And also in terms of the hypoglycemic response, is it merely the sympathoadrenal response and the local norepinephrine response that happens at the adipose tissue, the muscle, the liver? Are those happening independently, or are those part of one individual function that just breaks off those and I think that is the really interesting part. If we can correct individual functions or target them all together, and just how are those organized? I think that's a fascinating question that we have. And how are those also separately impacting feeding? Because I know also in some of the recent work is that the VMH also controls feeding responses as well, and how that differs from the regulation that happens within the arcuate nucleus. Because that's something that I have thought of a lot, and that puzzles me, is that why would you have these functions in a place like the ventral medial hypothalamic nucleus just above the arcuate nucleus? What is distinct from the regulation that happens there versus that in the arcuate? Certainly there are differences between the function there, but there are a lot of similarities between those areas. And why would one evolve in one way and one evolve in the other? And I don't have a great answer for that. But I think potentially in understanding the differences and the similarities between them could provide some context to overall metabolic function.
Yeah, so many very interesting questions there. Thank you, Dr. Flak, for joining us today. And thanks to our listeners for tuning in to this episode of Sweet Talk. Be sure to check out Dr. Flak's article titled Functionally Separate Populations of Ventromedial Hypothalamic Neurons in Obesity and Diabetes, available in the January 2025 edition of Diabetes. And as always, please like, follow, and subscribe for more Diabetes Bio content.
Thanks to all of you for joining us on the Diabetes Bio podcast. We hope that you're enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature articles we discuss in each episode are freely accessible at diabetesjournal.org/diabetes and are also listed in the episode notes for your reference. As with any podcast, it really helps us if you like, rate, follow, and subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you each month.